Across a causeway, in the middle of the Firth of Forth, lies the ghost island of Cremond. Cremond Island is a desolate tidal island about 1 mile out to sea, which is connected to the mainland at low tide across the Drum Sands. The island is made of microgabbro, formerly known as dolerite, a subvolcanic intrusive igneous rock. A paved path, exposed at low water, allows access. This causeway runs at the foot of a row of concrete pylons on one side of the causeway, which were constructed as an anti-boat boom during the Second World War. At high tide, the path is covered by several feet of seawater, which cuts the island off from the mainland. Any visitors ensure that they leave enough time to return to the mainland before the water rises. The swiftness and speed at which the turning tides of the Firth of Forth come in can easily trap the unwary. The water levels rapidly become deep, immersing the pathway and severing any connection to the mainland. The mile-long causeway itself is a spectacle. It is lined with colossal concrete teeth designed to stop boats sliding through at high tide. The row of identical concrete pylons creates an imposing and arresting, if accidental, piece of brutalist architecture. Fissured, wrecked, dismembered, and fragmented here and there where a pylon has collapsed, succumbed, and fallen to the waves. At high tide, the concrete pillars are nearly submerged. The landscape is at once wild, dynamic, and vulnerable. Scotland suffered more than 500 German air raids during the course of the Second World War. Scotland was a strategic interest point for Hitler due to its coast and many naval sites, one of these being Scapa Flow in Orkney, which was the main British naval base during World War II. At the outbreak of World War II, Cremond Island, along with other islands in the Forth, was refortified and armed, designed specifically to tackle fast-moving torpedo boats. An anti-submarine net and anti-boat boom was laid across the estuary from Cremond Island directly to Inchcombe and then to the Charles Hill Battery on the Fife coast. The barrier was to protect ships in the anchorage from attack by torpedo boats and to stop submarines entering the anchorage to attack shipping or to damage the dock gate of Rosyth Dockyard. The line of concrete pylons was built from Cremond Island to the shore to complete the anti-boat barrier. The island itself was used as part of the River Forth's secondary defense line. The deserted island is dotted with wartime ruins. The causeway is slick with seawater and the green of slippery, slimy, glossy and gleaming algae. The tide recently having receded to a secure remoteness. The concrete pylons surveil us as we cross the causeway, sentinels on shore, custodians or decomposing teeth of the sea. The decay draws into focus the loss of use value, where the object's worth is realized only in the process of consumption, and the disintegration and dissolution of the aura. The ruin is at one level an object, at another a process or an action. The crumbling, corroding, putrefying power of nature slowly creates entirely new, meaningful forms. Nature has now made it her own. The concrete ruin is consumed, engulfed, and appropriate creatures revel. The ruin is a constellation of meanings that move between past, present, and future. The ruin is a reminder of the universal reality of collapse and rot. A warning from the past about the destiny of our own or other civilizations. A fetish or object of desire. A topography of insatiable greed and profit. The inferno of the living, easily forgotten in the midst of history's terrible repetitions. Transformed into a corpse. A monotonous inventory of parts that are crumbling. The very picture of economic hubris or industrial decline. An ideal of beauty that is alluring exactly because of its flaws and failures. A dissected body, the symbol of a certain melancholic or maundering state of mind. A scar, a silent witness. An image of equilibrium between nature and culture. A desolate playground in whose cracked and weed-infested precincts we have space to imagine a future. And most unsettlingly perhaps, the ruin conjures a future past, the memory of what might have been. We ask a great deal of ruins and divine a lot of sense from their silence. Everything comes to nothing. Everything perishes. Only time endures, no matter how fractured or dissolved at its edges. In the zone, nature and culture, landscape and ruin begin to bleed into one another, so that we can no longer truly say what is ruin and what its background? What is monument and what the dead thing it recalls? I felt that the decrepit state of things precisely reflected my own state of mind. It was cold out on the causeway, with intermittent rain showers and a blustery wind. We stayed there for a while, incapable of leaving, held like the water as it enters the drift of reeds, unhurriedly thickening, transformed. I look back at the mainland from the island. The edge of the sea is a strange and beautiful place. For no two successive days is the shoreline precisely the same. The tides advance and retreat in their timeless rhythms. The sea is never at rest, never at ease. Living material is collected from diverse environments of the zone, including soil, water, air, and biofilms. With time against us, we decided to retrace our steps and head back to the mainland. The tide was coming in fast, the waters of the Firth of Forth rising steadily. The imposing ruins will soon be consumed by the sea, but only to unfailingly rise again. What lies beneath the surface and the soil? What enigmas and nightmares might this remote site be camouflaging? The zone. A region, area, stretch of land, or section characterized by some distinctive feature, purpose, or quality. An area subject to a particular political, military, or government function, use, or jurisdiction. A demilitarized zone. An area subject to particular restrictions. One of the divisions of the Earth's surface. A distinctive layer or region of rock characterized by particular fossils. An area, especially a belt of land, having a particular flora and fauna determined by the prevailing environmental conditions. A portion of a sphere between two parallel planes intersecting the sphere. A time zone. A girdle or belt. A sphere of thought, disagreement, argument. I think about the more than human life forms which inhabit tidal zones. I start to wonder, what does cohabitation mean in a time of mass human destruction? What is the relationship between capitalist destruction and collaborative survival within multi-species landscape? What is the prerequisite for continuing life on Earth? Back in the laboratory, we decided to construct devices for culturing a large diversity of microorganisms. Unique miniature microbial ecosystems or microbial gardens. The column is a rough mixture of ingredients. The glass containers are filled 1/3 full of mud, omitting any sticks, debris, and air bubbles. Supplementation of calcium carbonate and a sulfur source is required, mixed in with a carbon source containing cellulose. In the 1880s, Russian microbiologist Sergei Vinogradsky studied the complex interactions between environmental conditions and microbial activities using soil enrichment to isolate pure bacterial cultures within the column to gain an understanding of how they occur in nature. The structure of a microbial community is the result of environmental factors, evolutionary processes, and neutral or stochastic processes. Once prepared, the column is a self-sustaining, enclosed ecosystem dependent only on input of light as an exogenous energy source. Much like a gardener tending to his plants, providing the finest conditions for a plant species to grow, constructing a column provides a rich environment for microbes to grow or bloom as a lush population. An additional anaerobic layer, this time of unsupplemented mud or sand, brings the container to 2/3 full. Using sand for the layer above the enriched sediment often allows for easier observation and sampling of resulting populations. The column provides a whole range of environments in one small setting. A microcosm enabling many types of organisms with different requirements to grow in different sections of the column. This is followed by seawater to saturate the mud or sand and occupy half the remaining volume. The column is sealed tightly to prevent evaporation of water and incubated for several months in natural light under controlled laboratory conditions. Over time and space, microbial activity and abiotic processes result in chemical and environmental gradients from top to bottom and surface to interior of the columns, resulting in diverse niches for microbial growth and providing a unique opportunity to evaluate the development, alteration, and response of the microbial community. The prepared columns were observed over several months for development of layers, smell, colors, and zones. As the microbes in the soil photosynthesize pigments, we are exposed to the processes of growth and decomposition of various species of bacteria within this ecosystem, with variations in populations observed through waves of color. Incubating the column in available light for several months results in an aerobic-anaerobic gradient as well as a sulfide gradient. These two gradients promote the growth of different microorganisms such as Clostridium, Disulfovibrio, Chlorobium, Chromatium, Rhodomicrobium, and Beggiatoa, as well as many other species of bacteria, cyanobacteria, and algae. The column provides numerous gradients depending on additive nutrients from which the variety of aforementioned organisms can grow. The aerobic water phase and anaerobic mud or soil phase are one such distinction. Because of oxygen's low solubility in water, the water quickly becomes anoxic towards the interface of the mud and water. Anaerobic phototrophs are still present to a large extent in the mud phase, and there is still capacity for biofilm creation and colony expansion. Algae and other aerobic phototrophs are present along the surface and water of the upper half of the columns. Green growth is often attributed to these organisms. A microbiological culture is a method of growing and multiplying microbial organisms by letting them reproduce in predetermined culture medium under controlled laboratory conditions. Bacterial culturing is the process of growing bacteria. Culturing involves the making of sterile medium, inoculating, incubating, and examining microorganisms. Microbial cultures are grown in petri dishes that have a thin layer of agar-based growth medium. Agar, which is a polysaccharide derived from red seaweed, Rhodophyceae, is preferred because it is an inert, non-nutritive substance. A serial dilution is a series of sequential dilutions of a substance in solution. It is used to reduce a dense culture of cells to a more usable concentration. Usually, the dilution factor at each step is constant, resulting in a geometric progression of the concentration in a logarithmic fashion. Bacteria are a type of biological cell. They constitute a large domain of prokaryotic microorganisms. Bacteria were among the first life forms to appear on Earth and are present in most of its habitats. Bacteria also live in symbiotic and parasitic relationships with plants and animals. Most bacteria have not been characterized, and only about 27% of the bacterial phylae have species that can be grown in the laboratory. Bacteria were first observed by Anton von Leeuwenhoek in the late 17th century, but didn't become the objects of serious scientific study until the 19th century, when it became apparent that some species caused human diseases. The methods devised by Robert Koch, Louis Pasteur, and their associates during the golden age of microbiology, which spanned from the mid-1800s to early 1900s, are still widely used today. During the middle of the 20th century, bacteria became popular subjects of empirical study in fields such as genetics, genetic engineering, and biochemistry. With the evolution of antibiotic-resistant strains and our increased knowledge of bacterial stealth attack strategies, such as biofilms and intracellular growth, medical researchers have refocused their attention on disease-causing bacteria. Bacteria grow on solid media as colonies. A colony is a visible mass of microorganisms, all originating from a single mother cell. Therefore, a colony constitutes a clone of bacteria, all genetically alike. As the bacteria consume the nutrients, they begin to grow and multiply. This generates thousands to millions to billions of cells that begin to pile up, becoming visible to the naked eye. This pile of cells originates from one cell and is called a bacterial colony. Today, biologists face the conceptual challenge of rethinking bacteria as partners in health. Human bodies can no longer be seen as fortresses to defend against microbial onslaught, but must be re-envisioned as nested ecosystems. In the era of the Anthropocene, noticing microbial worlds seems more important than ever. Simply put, sustaining life requires sustaining symbiosis, and noticing these symbioses is a critical step. According to Professor Margaret McFall-Nye, New technologies are clearly a great help for seeing trans-domain partnerships. But we need more. We also need new collaborations among the different branches of biology and beyond. The postmodern synthesis is a theoretical frame that demands such disciplinary practices. However, it is also something more. It is also a general call for all of us to be more curious about the microbes that make up our worlds. The agar provides a solid growth surface for the bacteria, upon which bacteria reproduce until the distinctive lumps of cells that we call colonies form. Once the growth medium in the Petri dish is inoculated with the desired bacteria, the plates are incubated at the optimal temperature for the growing of the selected bacteria. Working with biological processes and structures can be both challenging and provocative. Scientists and artists often collaborate together in what become teeming, innovative spaces of co-creation. However, creating and working with lively material in the laboratory brings with it a range of ethical, social, cultural, technical and aesthetic inquiries. I find myself provoked with these thoughts, and thoughts are powerful things. What constitutes life? What counts as a sentient being? What does creativity mean in the context of the laboratory? Who gets to determine what lives are created and grown, which are saved, exploited, or destroyed? What do we think about using living materials such as live tissues, bacteria, living organisms, and life processes as a medium? What does it mean to have agency and ownership over another's life? Do we think of this lively material as a carrier, holder, vessel, receptacle, or repository that is populated by our own thoughts and ideas? What is the role of human beings as makers? What do we think about the disposal of these lively objects that soon become infectious laboratory waste? What does it mean to be at risk with each other? What are the possibilities and limitations of working with other than human lives? What do we think of contamination? Contamination. The action or state of making or being made impure by polluting or poisoning. The presence of a constituent, impurity, or some other undesirable element that soils, corrupts, infects, makes unfit, or makes inferior a material, physical body, natural environment. The fear of being endangered or changed by direct or indirect contact with certain types of people, ideas, or situations. Contaminant. A substance that makes something less pure or makes it poisonous. A contaminant may even be more abstract, as in the case of an unwanted energy source that interferes with a process. The evolution of our selves is already polluted by histories of encounter. They change who we are and make way for others. As contamination changes world-making projects, mutual worlds and new directions may emerge. We all carry a history of contamination. Purity is not an option. Staying alive for every species requires livable collaborations. Collaboration means working across difference, which leads to contamination. What strikes me is that we are all interconnected by shared ecologies, and entanglements with our microbial relations makes life probable. Entanglement is a fundamental aspect of all life. Yet we have more often than not been in common denial of these entanglements. But who are we, and what is ours? The I within us contains multitudes. The human microbiota is the aggregate of microorganisms that resides on or within any of a number of human tissues and biofluids. Those that live with us, the microbiome, are invaluable parts of our lives. We are less individual and more interconnected, interdependent multitudes. But do we recognize the importance of these symbiotic makings? Contamination makes diversity. Contaminated diversity is complicated, often ugly, and humbling. All entanglements and all symbioses are vulnerable, and relations with others must be continually re-energized and negotiated within life's frameworks. When circumstances change, these very life-sustaining equations turn awry. There is a frightening collision then between the possibilities and limitations of human and non-human life, caught as it were between nightmare and dream. So what values can emerge from ruined landscape? If if a rush of troubled stories is the best way to tell about contaminated diversity, then it is time to make that rush part of our knowledge practices. It is in listening to that cacophony of troubled stories that we might encounter our best hopes for precarious survival.